Hello students, I am Dr. Mahalakshmi. I am heading the Microbiology department at Balaji Dental College, and I have been teaching the dental students for the past 3 decades, and I also assist the PG students, MDA students for their project work, and we do a lot of interdisciplinary research with various departments like orthodontics, periodontics, prosthodontics and endodontics. And today we will discuss about history of microbiology. And most of the dental students don't show interest to this particular topic, but examination point of view, you all do get a short notes from this particular topic and also MCQs from this history of microbiology. And I am not going to discuss this particular subject with a timeline. We are going to see the important contributions of 5 different scientists who has contributed much to this particular field of microbiology. So, to start with, the person who first identified the microorganisms and reported. So many people observed these microorganisms earlier to Antony van Leeuwenhoek. Antony van Leeuwenhoek was the first person to observe and to report his findings. He was the first person to report his findings. In fact, his period was between 1632 to 1723. And during the main hobby of Antony van Leeuwenhoek is to grind lenses. His hobby was to grind lenses. So when he was grinding lenses, he was able to see small animalcules moving. And the lenses which he used during his period, we didn't have any advanced microscope during his period, and it was just an hand lens which a palmist used actually, and that magnified around 200 times. With such low magnification, he was able to picturize beautifully the morphology of these microorganisms, especially the bacteria. And he even gave the sketches of these bacteria. Some are spherical shaped, some are rod shaped, and some are comma shaped, and some are spirally coiled. And the sketches which he has given, and that too in his period is between 1632 and 1723. There is no change even after advancement of microscopes. Even today we have the microorganisms and especially bacteria showing spherical shape, rod shaped, and spirally coiled and comma shaped bacteria. So since he was very much interested in microscopy and he was the one who observed and reported, he is referred the father of microscopy. Antony van Leeuwenhoek is referred the father of microscopy. Next we will see the contributions of Louis Pasteur. Louis Pasteur basically is a French chemist, and his period is between 1822 to 1895. And being a chemist, he was more interested in fermentation reactions and all that. And during his period, actually you can see Antony van Leeuwenhoek, he reported the findings before 1723. And nearly for 200 years, there was no improvement in this particular science. And during Pasteur period, there was a big debate between two group of scientists. One group of people believed these microorganisms. They had a big question how these small animalcules originated. And one group, they kept on telling it is by abiogenesis, from a dead animal or meat particles, these microorganisms arises. But there was one more group of people who disbelieved it, and they said mainly they originate from a parent cell. So they strongly believed on biogenesis. Biogenesis is development of any organism from a parent cell, from a living cell. So there was a big debate going on. And so many people worked with so many experiments, and to note some are really notable experiments, especially the one which was given by John Needham. What he did, he heated the broth, he cooled it and sealed the flask, and then he observed the broth. He was not able to see the small animalcules, but still the people who were strongly believing on abiogenesis, they were not convinced. They said air is necessary since the sealed flask, the flask was sealed, they were not able to see the bacteria. Later on, Lazarus Palensy heated the broth for a long time, he didn't seal it, but again he was not able to, the people were not convinced by his experiment also. They kept on telling this particular heat has destroyed the microorganism. Later on, Franz Schulz and Theodor Schwann, they did 2 experiments, one with the acids, another one with heat. They kept the meat broth in a flask, and one person passed the air through the chemical, another person through hot tubes. Again, the people who believed on abiogenesis was not convinced. They kept on telling the heat and acids has spoiled the environment and they were not able to see the microorganisms. And this was a big breakthrough. wherein Pasteur showed a lot of interest on this, and somehow he wanted to disprove the spontaneous generation. And for this purpose, the gooseneck flask was very, very helpful. So he took a gooseneck flask. Hope you all know what is a goose. It's a bird with a very long neck. And he took a flask similarly, and he placed the cooked broth in this particular flask, and he left it open for a few days, 5 to 6 days. And after a week, he opened it and he took the broth. When he observed, he was not able to see any animalcules. And just this time, the people who strongly believed on abiogenesis, they agreed with Pasteur's finding, and Pasteur was the one who disproved the spontaneous generation. And how did he was able to prove this with a gooseneck flask? The principle behind this is he didn't seal the flask, but the long neck, curved neck, actually, when the air enters in through this long neck, the pressure is reduced. In the reduced pressure, the microorganisms or any particle which is carried along with the air gets deposited in the neck of the flask. So only the sterile air enters into the flask. So this was the principle behind this experiment, and the people who believed on abiogenesis were strongly convinced, and they accepted Pasteur's experiment, and it was a big finding by Pasteur, a contribution to this field of microbiology. So he was the person who disproved spontaneous generation. So he gained a lot of fame during this period, and again during Pasteur's period, the wine industries faced a lot of problem. They were not able to sell the wine got spoiled, it gave a very bad odor, and it was bring a heavy loss to these industries, and they all approached Pasteur and they wanted to identify what was the reason for the spoilage of the wine. So Pasteur, basically being a chemist, and he had a lot of interest on the fermentation reactions. Hope you all know how the wine is prepared. Wine is prepared by fermentation of grape juice using microorganisms, especially the bacterial species. So he did a lot of experiments, and he identified that the bacteria which were fermenting this wine, there were two batch of bacteria. One batch he referred it as bad batch, another batch he referred it as a good batch. The good batch of bacteria developed a good quality of wine, while the bad batch spoiled the wine, and that was the reason why the wine industries was facing a lot of problem. Now he identified the problem, and now he wanted to get rid of this bad batch of bacteria. And for this, what he did a lot of experiments, and he identified this bad batch of bacteria are actually thermolabile. Easily you can kill them at lower temperatures, while the good batch of bacteria are all thermophilic. They grow very well at higher temperatures. So what he did, he heated this particular wine product at temperature of 62 degrees centigrade for 30 minutes, and all these bad batch of bacteria were destroyed, and a good quality of wine was produced. And it was again a very good contribution to this field of microbiology. And to honor this particular contribution, the process of removing this bad batch of bacteria by killing at 62 degrees centigrade for 30 minutes was referred as pasteurization, to honor his work. And you all know this pasteurization is now also we are using it in food industries and dairy industry, especially to preserve the food, and also in milk and dairy industry, mainly to destroy the milk-borne pathogens. We are using this pasteurization. So in fact, you can see in the milk sachets, it will be written Padapadta Pattapal. It is nothing but pasteurized milk, where they have removed all the bad batch of bacteria, especially which can cause milk-borne diseases. So he gained a lot of fame in this field of microbiology, and he himself was more interested in this particular science, and he identified a vaccine for anthrax. Hope you all know anthrax is a zoonotic disease which is caused by Bacillus anthrax. Both human as well as animals can acquire this disease. Humans can accidentally acquire this particular disease, and he was the one. In fact, this bacteria was identified by Robert Koch, but vaccine was discovered by Pasteur. He found it a vaccine, and again it is really a boon for the medical field. The animals, the livestock were protected by giving this vaccine from anthrax. And another important finding and interesting finding of Pasteur is he identified a vaccine also for chicken cholera. And this is quite interesting how he identified this chicken cholera. And in those days, when any discoveries was done, they are supposed to demonstrate this experiment in front of a public. They call for a public meeting and they have to demonstrate it. The public should be convinced by the experiment. So he identified that there was one bacteria which was responsible for this chicken cholera, and during this period the poultry farms, they faced lot of problem, and they met a heavy loss. The birds died due to cholera. So Pasteur did a lot of experiments and he identified the etiology of chicken cholera, and he wanted to demonstrate in front of a public. So he brought the birds, chickens into the public meeting, and he also acquired the culture. He injected the culture in front of the public, but unfortunately the birds did not get affected with the disease cholera, and they were live, they didn't die. It was a failure. That demonstration was a failure. Then he went back to his laboratory, and he wanted to identify why in the lab he was able to demonstrate this, why it was failed in a public meeting. He did lot of experiments, and after 3 or 4 months again he called for a second public meeting, and he took the birds which he already used it in the earlier demonstration, and also new batch of birds. See how discoveries are always accidental. You can identify that in this particular experiment. So he took 2 batches of birds, and he took a fresh culture this time, and he injected it into the 2 batches, and the batch which already received the dose of this bacterial species did not die. They didn't acquire the disease, whereas the new batch of birds which received these injections, they died. So now it is known the one which he had given the injections earlier has started developing immunity to that particular bacteria, so they were not affected with the disease, while the new batch of bacteria which was receiving the first dose, they died. So in this there was 2 discoveries. One, he identified the etiology for chicken cholera. Another one is the method of attenuation, how you can inactivate the bacteria so that the bacteria do not produce the disease. At the same time, it is stimulating the immune system of the host to develop the antibodies. So he also found out the attenuation and the vaccine for chicken cholera. So this is another important contribution of Louis Pasteur. And he also coined the terms vaccine and vaccination. And that I will later on discuss when we see the contribution of Jenner. He only coined the term vaccine and vaccination, Pasteur. And he also worked on various diseases of silkworms and how to control the diseases of silkworms. And another important discovery of Louis Pasteur is he discovered a vaccine for hydrophobia, that is rabies. It was really a big breakthrough, and I hope you all know this particular disease, rabies, is caused by a virus. But during Pasteur period, when he discovered a vaccine, they didn't know the etiology because they didn't have a electron microscope or not even a light microscope. So they, without even knowing the etiology, the vaccine was discovered for rabies. And even today, we can prevent the disease, but we cannot cure the disease. Hope you all know it is not a curable disease. Rabies, you can only prevent it. And I think we should thank Pasteur for such a beautiful contribution to this field of microbiology, rabies vaccine. And how he discovered, that is quite interesting. He found that a person who had a bite with a rabid dog, they died. And he was just keenly watching. Since they had a bite by the rabid dog, must be the saliva should be the culprit, which must be having these microorganisms. So what he did, he took the rabid saliva from the rabies dog, and that time he used rabbits for his experiment. He injected this saliva intracerebrally into the rabbits, and he allowed the particular agent to multiply in the rabbits for about a week or 10 days. Then he sacrificed the animal, he cut open the head of the animal, removed the brain, he homogenized the brain, and he purified it, and he was preparing a vaccine. And during this time when he was preparing, a boy by name Joseph Meester, he was bitten by a rabid dog. So he took permission from his parents, after ethics consideration from their parents. He gave this vaccination to the boy, and the boy was saved from rabies. This is how he discovered a rabies vaccine. This was in the year 1885, July 6th. He was the first boy to receive the vaccine for rabies. And nowadays we don't prepare with these rabbits. In fact, in King Institute, Guindy, some 15-20 years back, they used to prepare it in sheep's brain. Of course, now they have stopped completely preparing any vaccines with the animal model because of any allergic reactions that we can develop due to the animal products. So now genetic engineering methods are followed for developing the vaccines. And if you see Pasteur's contributions, he would have contributed in different branches of microbiology like general microbiology, immunology, virology, bacteriology. So since his contributions was in different branches of microbiology, he is revered the father of microbiology. So this can come as a short note in your examinations, BDS examinations. Contributions of Louis Pasteur. Next, we'll move on to contributions of Robert Koch. Robert Koch period is between 1843 to 1910. Basically, he is a German bacteriologist. And he was the first person to introduce stains and staining methods. We all know the bacteria can be visualized easily under a microscope even without staining. But the clarity may not be very clear. So he was the first person to introduce stains and staining methods in bacteriology. And another important finding of Robert Koch is he identified agar-agar, a substance used as a solidifying agent. Just imagine his period is between 1843 to 1910. No substance has replaced this agar-agar even today. It's a best solidifying agent. This agar melts at 80 degree and solidifies at 40 degrees. See, this can be an MCQ question. This agar, melting point of agar is 80 degree, solidifying point of agar is 20 degree. So this is agar substance is actually it is derived from a seaweed Gelidium. From this only this is produced. It's basically a solidifying agent. It's a big contribution of Robert Koch. But before this usage of agar-agar, before Robert Koch identified this agar-agar, we were using gelatin. Again, there was a drawback with gelatin at 37 degrees centigrade, that's our body temperature, it actually liquefies. And so no point in using that as a solidifying agent because we are going to incubate all the bacterial cultures at 37 degrees centigrade. So this was the best option which was introduced by Robert Koch. And another important contribution by Robert Koch is postulates. We refer it as Koch's postulates. So even Pasteur followed his postulates when he was discovering any etiological agent. Even today, if any person is going to identify an etiology, we have to fulfill this postulates. You have to demonstrate it by Koch's postulates. So to identify any causative agent of an infectious disease, the methodology that was put forth by Robert Koch. So 4 criteria was established by Robert Koch to identify the causative agent of a particular disease. The number 1, the microorganism or the pathogen involved should be present in all cases of the disease. Number 2, the pathogen can be isolated from the deceased wound in the laboratory as a pure culture. The third, the isolated pure culture, the pathogen, when introduced into a susceptible animal, the animal should show the same clinical signs and symptoms. The fourth one is the pathogen can be re-isolated from the susceptible animal and as you observe it originally from the human host. These are the 4 important criteria which was put forth by Robert Koch. The 5th one was added now to this postulates, that is the animal and the human serum will contain the antibodies in their serum towards the specific pathogens, against the specific pathogens. So it's a very, very important Koch's postulate. So this can be even asked as a short note or in a contribution. If you are in a short note, if Robert Koch's contribution is asked, this alone postulates will be given 2.5 marks. So this slide just shows you the pictorial form of Koch's postulates. So here you could see the animal cow is suffering with anthrax, and from the animal, dead animal, you are drawing the blood and you are inoculating it into a culture and you are growing this organism. After you grow it, you identify it by microscopy and other tests. Then you take the pure culture, when you inject it into a healthy animal, now the healthy animal also will show the same clinical signs and symptoms. And Robert Koch was the first person to use laboratory animals for experimental infections. He was the first one to use laboratory animals for experimental infections, and he also discovered the etiological agent of tuberculosis, that is Mycobacterium tuberculosis, and the etiological agent of cholera, Vibrio cholera, and anthrax bacillus. All these 3 was discovered by Robert Koch. So when you just see his contributions, Koch's contribution, it will be mainly in the field of bacteriology. So he is referred the father of bacteriology. And another important note, Robert Koch received a Nobel Prize in the year 1905 for discovery of tuberculosis. You may wonder, how come Robert Koch received a Nobel Prize? Why not Pasteur? If you see the period, Pasteur's period is actually till 1895, but Nobel Prize was introduced only in 1901. So that is the reason Pasteur didn't receive the Nobel Prize, but actually especially he should have received a Nobel Prize for his contributions for vaccine for rabies. So this was the reason why he couldn't receive the Nobel Prize. It was introduced only in the year 1901. Next, we move on to contributions of Edward Jenner. Edward Jenner was even before Pasteur period. His period was between 1749 to 1823. Basically, he is a English physician. And he was just observing, hope you all know about this disease, smallpox. Even though it is referred as a smallpox, in Tamil we refer it as periammai. We don't call it as Chinnamai, we refer it as periammai. You would have seen your grandmas, and in that particular generation how the people would have fought this disease, and some who survive after this disease, there will be some morbidity in them. Some would have lost their eyesight, some would have become deaf, some had worst scars on their face. So these are all morbidity of this particular dreadful virus, smallpox, and we think we all should thank Jenner for identifying a vaccine for this particular disease. This is the one and only contribution of Jenner, but it is an excellent contribution to the field of microbiology. And how he discovered this? He was just observing the maids, especially the milkmaids. They didn't acquire the disease, while other populations suffered from this particular smallpox. So he was keenly observing why come these particular people did not acquire the disease. But he identified small pustule vesicles on these milkmaids in their fingers, and this was due to a virus called cowpox virus. A cowpox pustule was there. So what he did, he took the material from these milkmaids' cowpox lesions, and he wanted to use this as a vaccine. And again, at that time, a boy named James Phipps, he only received the first vaccine, smallpox vaccine. He took the permission from their parents, and he was injected with this cowpox material. And after a few weeks, when he was again exposed to smallpox virus, this boy did not acquire the disease smallpox. So he was the first person to receive, and it was really a very, very big boon to the medical field. And then it was mandatorily given to all population, and during some 40-45 years back, the primary health workers used to go to each and every one's house and see to that every individual is vaccinated against this particular disease. And in the year 1980, the World Health Organization declared this disease as an eradicated disease. Smallpox is now eradicated totally, so we don't have this vaccine in our immunization schedule, and we all should thank Edward Jenner for this contribution. So completely this disease has been eradicated. Hope you all know now we are similarly fighting for one more disease to eradicate, that is polio. Every year we are having these pulse polio programs, vaccination for 0 to 5 years children in the month of December and February. Maybe in another few years, WHO may declare this particular disease also as an eradicated disease. So this was the only contribution of Edward Jenner. Earlier I told you Pasteur coined the term vaccine and vaccination. This is just to honor Jenner's work, because Jenner used a cowpox material. In Latin, vacca means cow. So to honor Jenner's work, Pasteur coined this term vaccine. Vaccine is a substance which is given to prevent any disease, and vaccination is a process of giving a vaccine. So these two terms was actually coined by Pasteur to honor Edward Jenner. Then we will move on, and this is the picture just shows you Edward Jenner vaccinating James Phipps with cowpox material. And another important note regarding the smallpox virus. So any particular living, right, even though it is a unicellular which you cannot visualize through your naked eye, but still no living creature should go extinct. So even though we have eradicated the disease, the organism has not gone extinct. It is still officially living in 2 repositories in the planet, one in Center for Disease Control and Prevention, Atlanta in USA. Another one in Russian Virology Institute called Vector. And in these two repositories, the virus is staying alive with tight security, because there will be a scare whether the people will be using this as a biological weapon. So what will happen if they use this as a biological weapon? Vaccination has been stopped completely in the year 1980. People who are not vaccinated are at risk of acquiring this particular disease. So we may not know in future any viruses resembling a pox virus, smallpox virus can originate, and that time we may not have this virus to do any research on it. So that is why, since it should not go extinct, it is stored in these two repositories. Next, we will move on to contributions of Joseph Lister. Joseph Lister's period is between 1827 to 1912. He is basically an English surgeon. He was the first person to introduce antisepsis in surgery. Basically, in those days, during this Lister's period, they used to perform the surgery in a hall, huge hall. Nowadays, you have an excellent operation theatres where you can completely eradicate all the microorganisms, you sterilize it, you can fumigate the room so that the infections are prevented. But during his period, we were not aware of this antisepsis agents. So he used to perform surgeries, and most of the surgeries used to fail. Nearly about 80 to 85% who has undergone surgery can go for mortality usually. So he was wondering, surgery would have been done normally, but why these people died? So then he recognized there was certain infections which arised in these particular patients. That was the problem for a death. So what he did, he used carbolic acid as an antiseptic agent. So carbolic acid is nothing but phenol. He diluted this and he used to paint the area of the skin where he used to perform the surgery. And he found out, after using this method, almost 99% of death was prevented, mortality was prevented. And he is referred the father of antiseptic surgery. In the year 18, as a dental students, you should know about this. In 1879, to honor Lister, the mouthwash is named as Listerine mouthwash. The chlorhexidine mouthwash was given his name, Listerine mouthwash, to honor his work in the antisepsis. And this is the only contribution of Joseph Lister. So we'll move on to contributions of Alexander Fleming. His period is between 1881 to 1955. Basically, Fleming is a Scottish bacteriologist, and he is the one who discovered lysozyme. That was in the year 1922. So any students, if we ask contributions of Alexander Fleming, immediately they remember only penicillin, but also remember his contribution about lysozyme. That was in the year 1922. He is the first person who discovered this enzyme. Hope you all know this. Lysozymes are produced by lysosomes, which is referred as suicidal bags. It is responsible for autolysis of any cells. And in the year 1928, he discovered the antibiotic penicillin. Again, it's very, very interesting to see how he discovered this antibiotic. Basically, being a bacteriologist, he was working with a lot of bacterial cultures. So one day he just placed a culture containing Staphylococcus aureus in his drawer. And after a week, he came and observed these plates. He was not able to see Staphylococcus aureus. Instead, some other mold-like growth was seen surrounding the area where the colonies were present. And this mold-like growth was nothing but Penicillium notatum, the fungi. So a substance this produces, which is showing an antibacterial effect on Staphylococcus. So then he identified this was the substance which killed the Staphylococcus, and he named that particular substance as penicillin, basically because it was produced by the fungi Penicillium notatum. This was the first antibiotic to be introduced in the year 1928. So during that period, it was a miracle drug. Almost for any infections, they used to give penicillin, and it also works beautifully. But nowadays, we cannot use this penicillin because drug resistance has developed in bacteria. Almost to all drugs, this bacteria has developed resistance, and penicillin has now become a narrow narrow choice in using this penicillin as an antibiotic. So this miracle drug penicillin was discovered by Alexander Fleming. For this discovery, he received a Nobel Prize in the year 1945. With this, I conclude this topic, History of Microbiology. And if you have any clarifications, you can reach us.